Please explain in detail biology, evolution on Hardy-Weinberg equilibrium, and data analysis on experiment 1 and 2. Biology evolution, Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium, HWE, is a fundamental principle in population genetics that provides a baseline to study genetic variation in a population. It was independently proposed by G. H. Hardy and Wilhelm Weinberg in 1908. HWE assumes that the allele and genotype frequencies in a population remain constant from generation to generation, unless specific factors, e.g., mutation, selection, migration, genetic drift, or non-random mating, influence them. The HWE is based on the following assumptions. 1. Large population size. Genetic drift is minimized, and allele frequencies remain stable. 2. Random mating. Individuals in the population mate randomly, and no preference is shown for specific genotypes. 3. No mutation. No new alleles are introduced into the gene pool. 4. No migration. There is no gene flow between different populations. 5. No selection. All individuals have equal chances of survival and reproduction, and no specific genotypes are favored. Under these conditions, the allele frequencies, P and Q, and genotype frequencies, P squared, 2pq, and Q squared, remain constant over time. Here, P is the frequency of the dominant allele, A. Q is the frequency of the recessive allele, A. P squared represents the frequency of the homozygous dominant genotype, A. 2pq represents the frequency of the heterozygous genotype, A. And Q squared represents the frequency of the homozygous recessive genotype, A. Data analysis. Experiment 1 and 2. In these experiments, 2 bacterial strains, 1 and 2, undergo identical incubation on 3 plates with different starch agars. The starches are separately distributed within the agar, providing different environments for the bacteria to grow. Experiment 1. Growth of strain 1 on different starch agars. To analyze the data, measure the colony size or the number of colonies formed on each plate after a specified incubation period. Compare the results across the 3 different starch agars to determine if any particular starch type promotes or inhibits the growth of strain 1. Perform statistical tests such as ANOVA, analysis of variance, to analyze the differences in growth between the different starch agars. If there is a significant difference, conduct post hoc tests to identify which specific starch agars show significantly different growth patterns. Experiment 2. Growth of strain 2 on different starch agars. Follow the same procedure as experiment 1, but with strain 2. Compare the results across the 3 different starch agars to determine if any particular starch type promotes or inhibits the growth of strain 2. Comparison of strain 1 and 2 growth on different starch agars. To further investigate the growth patterns of strains 1 and 2 on different starch agars, compare the data between the 2 strains. This comparison can help identify differences in their metabolic capabilities, adaptations, or preferences for specific starch types. Perform statistical tests to determine if there are significant differences between the 2 strains in their growth on the 3 different starch agars. In conclusion, the Hardy-Weinberg equilibrium provides a foundation for understanding genetic variation within populations by assuming constant allele and genotype frequencies in the absence of evolutionary forces. In the experiments, comparing the growth of bacterial strains on different starch agars allows for the identification of metabolic preferences or adaptations in each strain. Analyzing the data using statistical tests can help determine if there are significant differences in growth patterns between the strains and across different starch environments.